Dr. Georgantas, you were just talking about some LNP, mRNA and LNP manufacturing, I guess paradigms, and I want to move sort of into that part of the conversation. I want to talk manufacturing challenges and opportunities, and let's start there. What are some of the, I guess, challenges, opportunities? How are you addressing them in terms of LNP quality and production technologies? And while you, I love these compounded questions, as you may have noticed. While you're responding to that, Robert, we did get an audience question. What's the current situation in terms of availability of development and manufacturing capacity at CDMOs for mRNA and LNP? Is there overcapacity, undercapacity, or just the right amount? So while you're responding to sort of some of the general manufacturing challenges, if you could work in a little outsourcing angle.
Well, I'll actually start with the outsourcing, because it's funny, because there's both, because of the COVID pandemic and the area being hot, and companies like McKinsey talking about just the CDMO market for mRNAs being a $28 to $30 billion market by 2029. It's an interesting question to answer, because there's both overcapacity and there's undercapacity at the same time, because we have a lot of companies, a lot of CDMOs that are jumping into the space for both the drug substance, what we call DS, which is the mRNA itself, as well as the drug product, which is the whole mRNA LNP construct, right? Mm-hmm. And what we're seeing across the industry now is that things are shaking out. There are some some CDMOs that are extremely good at this. There's some CDMOs that are claiming that they can do X, Y, and Z, and they can do everything up to pandemic scale, and they're new to the game and really are not experienced enough in this area to produce good product. They don't have maybe they were a biologic manufacturer that's, claiming that they can do mRNA LNPs, and it's a very different product, right? So, and that's why I say that you're seeing that there's a lot of capacity, because there are a lot of people that are claiming that they can do this, but there are only a handful of, CDMOs that are really good at this. I mean, we're actually at Providence, we're taking a step back, and, for everything, phase 1 and phase 2, we're actually building out our own GMP facility at the moment. Because through the pandemic and through the years of experience that we've had with making these constructs, we're quite good at it. So we're stepping back and bringing a lot of that back in house, for everything up to phase 2, and then currently teaching a select number of CDMOs, who I won't name, how to do this well and how to do it the right way. Because the real challenges here are, is that the things that we're seeing across the board, is that you really need new and robust analytic techniques, to assure that both your drug substance and your drug product are GMP quality and ready to go into a patient. And then the other thing that we're doing a lot of work, working out is the stability across the entire manufacturing pipeline, because one of the things that you do see with LNPs is that they're depending on which cationic lipid you're using, which excipient lipids you're using, and anything that you're adding in as a modifier to that system, is that they act very, very differently. You can, if I go back to the biologic realm, the monoclonal antibodies, you put a construct into a CHO cell, your challenges there are how well it expresses, but that technique's been worked out very, very well. And so, you're going to put a construct in, you're going to choose your clones, you're going to take one that's high expressing, and you have really good techniques for for purifying those antibodies. We're at the point that the field's early enough that we're still, you change the composition of your LNP a little bit, and it acts very, very differently, and that's something that you're going to have to work on a case-by-case basis. And it's something that, outside manufacturing organizations are going to have a problem with, is that there's not going to be one set pipeline. It's probably for each lipid, at least in the short term, it's going to be a custom pipeline for each lipid composition nanoparticle that you're making. And that's really the biggest challenge at the moment from my perspective.
Yeah. Peter, what are your thoughts on that? Are you seeing some of the same challenges in terms of outsourced, not just capacity, but ability?
Yeah, absolutely. So what we're often looking for is flexibility. So if you're a biotech company starting up, or if you're an academic group that moving into clinical, what you're looking for there is you're rolling a dice when it comes to CDMOs. You really don't know too much. It's difficult to do the complete diligence. I agree with Robert there that there's overcapacity and undercapacity. We were extremely lucky, I would say, in our case, but we had also, in our case, we also had some background from previous companies, and so that when we started HDT, we already had really good relationships with CDMOs. But what we often see, as Robert alludes to there, is this good capacity at making the drug substance and even drug product. There's an undercapacity when it comes to rapid, flexible, small-scale production. I would say that's my experience. So now, if there is someone out there who has a facility that very rapidly and at very small scale, but quickly make GMP product for personalized use, then I think they should reach out. But joke aside here, but we also see a rapid race from the COVID towards scale, perhaps not on the fill finish side as much as on the bulk mRNA drug substance. We use a slightly different LNP than Roberts and Pfizer and Moderna for that matter. We use an LNP or a nanoparticle that is on the inside. We actually wrap the RNA on the outside. Benefit there is that we can produce it separately and stockpile it. And our final vaccine product, which we call it a drug product, is what Robert would call it, is simply an admix of that can be produced either made either at the clinic pharmacy or in the manufacturing facilities. That by having a niche product like that, we bypass or we avoid having supply constraints, for example. We also don't need to ship things between facilities. It makes it easier for us to make ultra-personalized vaccines for us, and any type of vaccines for that matter, at low cost.
Yeah. Okay. I want to, we'll jump back into the mRNA space. We had a few questions around that, but I want to jump back over to Jo for a minute and get her perspective on therapeutic manufacturing challenges and opportunities. So just give us, I guess, the high level on that. What are some of the challenges and opportunities you're seeing from a manufacturing standpoint? And the outsourced angle from you might not be a bad commentary as well.
Yeah, sure. I think as a small biotech, similarly, we have obviously been relying in the CDMO space a lot. I will say our technology relies on tapping into decades worth of experience of manufacturing of live attenuated viral vaccines, which obviously have been out there and have been commonly used. And certainly we benefit from the experience in that place, both from an analytical as well as the upstream and downstream processing perspectives. I will say, though, that we have been struggling, I would say, a little bit with CDMOs that actually really have the right type of expertise, who have done and worked with particular viral species. Using one and manufacturing one viral species is not the same as another. Obviously, there has been a lot of experience gained in the CDMO arena related to lentiviral vectors or AAVs or adenoviruses, which are viruses that are used for gene therapy or other applications. But manufacturing different type of viral species, sometimes boutique viral species, is a different ballgame, and the right type of expertise is needed. So I would say in the virotherapy space, overlaying concerns around cross-contaminations, because we're dealing with live infectious virus here, whether real or perceived, overlay, complexities when it comes to closed systems, filtration not necessarily be an option for sterility measures or concerns at the end. So therefore, closed systems and the supply chain components that come along with single-use systems really, I think, create a layer in general for this type of modality for us that I would say there are not enough CDMOs out there with the right type of expertise and general relatively long lead times. So in that sense, I echo the concern around nimble and fast small-scale production runs for early phase trials that I would challenge the audience to help us overcome.
I like the challenges being thrown down here. You know, from my perspective as someone who is a journalist in the space and covers biomanufacturing, judging by the volume of square footage of outsourcing space cited in the press releases that come across my desk, like on the daily. I mean, you know, you would think that they're just expanding by the millions per minute. It's surprising to hear that there's sort of still a void to be filled in terms of outsourced manufacturing ability and capacity there.
Yeah, I think it's about the right experience, and certainly the virotherapy field often therefore takes the route to bring manufacturing in-house, which is obviously a large burden for the biotech space, which is generally where virotherapy development happens at this point in time. And it is quite risky from a financial perspective and otherwise to bring such a high investment capability in-house. But that is how many larger players, Replimune, for example, have addressed these concerns.